February 23, 1997, one of the century's greatest scientific achievements was unveiled before the eyes of a stunned world. Her name was Dolly the sheep. Scientists at the Roslin Institute near Edinburgh, Scotland, had created the first successful clone of an adult mammal. Their achievement had profound implications for society.
A Church of Scotland report has called for an independent commission to be set up to examine the ethics of biotechnology.
The announcement of Dolly's existence ignited a firestorm of controversy over cloning's possible uses and abuses. Even the President of the U.S. felt compelled to comment on the implications of this startling breakthrough.
Each human life is unique, born of a miracle that reaches beyond laboratory science. I believe we must respect this profound gift and resist the temptation to replicate ourselves.
Previously, most experts had believed the creation of a clone from an adult mammal was impossible, the stuff of science fiction. Clones had been created before, but they had been engineered from embryo cells, or from the adult cells of plants and non-mammals. Dolly represented a major scientific advance. Researchers had never before successfully used a cell from a fully grown adult mammal to make a genetically identical copy or clone of that mammal. To create Dolly, scientists at the Roslin Institute first extracted a cell from an adult sheep and then grew the cell in the lab for 6 days. The next step was to take an unfertilized egg from a second sheep, remove the DNA from that egg, and combine the egg with the cell from the first sheep. The resulting embryo was then placed in a third sheep. This third sheep gave birth to Dolly, an exact genetic copy or clone of the first sheep. The potential benefits and dangers that could come from producing exact genetic copies of animals, possibly including humans, were staggering. Dr. Ian Wilmot led the research team that created Dolly.
I don't think we can begin to estimate what new understanding this will offer, not only for normal health, but also for human diseases. And I think that our successors undoubtedly will see that that use will be very important.